Hello, my name is Nikos Leodakis.
I am Kelton Howell.
I'm Megan Dolly.
And we are all uprising seniors in Utah at Hillcrest High School in the Canyon School District. And the title of our project was The Effect of Microgravity on the Diffusion of Morphogens in Xenopus Laevis.
Okay, so how we all began. Our physics teacher introduced us to this whole project, the SSEP. And so we started researching projects. We wanted to find out what we could test in microgravity. So our biology teacher reminded us of the topic of morphogens. So we began researching this topic in depth and found a lot more stuff out about morphogens than we already knew. Morphogens are proteins in embryos. They are present in all embryos, so this project could be very related to humans as well as the frogs that we tested it on. They traverse tissue by simple diffusion across different nodes in embryos, and this is how the embryos know what to develop into because of the different concentrations of morphogens across the embryos. Activin is a specific morphogen in Xenopus laevis, and it is involved in mesodermal development, which would be the brain and spinal cord. So specifically, we were trying to see how Xenopus laevis developed in microgravity in regards to the spinal and brain development. We did research and found that previous experiments flown on Mach 6s and Mach 3s found different development in the brain and spinal cord regions. There was enlarged brain and curved spinal cord, which indicated that we may also be able to find some results.
So furthering with our background research, what we wanted to do is to figure out what we could do once we got this experiment. And so the purpose of our experiment was, with human involvement, all these advancements, we have species that are becoming endangered because of pollution, because of diseases, and these problems are affecting individuals. And so if we are able to use microgravity to change this concentration in these animals, we could change things like stopping abnormalities in animals and having the capabilities of enhancing traits so that they are better provided for survival and so that they can live in such a changing environment as we have today, because natural selection has had all these problems. And so that was the main purpose of using morphogens and activin specifically, to see if we can change these concentrations.
And when we designed our experiment, we based the question on how does microgravity affect the diffusion of morphogens in Xenopus laevis. Our independent variable was the microgravity conditions. The dependent variable was the diffusion of the morphogens. For our control, we left 2 eggs here on Earth under the same conditions, and our constants, and that's how we kept our experiment constant. Our hypothesis was, if microgravity inhibits the diffusion of morphogens, then the Xenopus laevis will develop abnormally. Our apparatus was set up in a material dispersion apparatus, and we had 2 frog eggs, one in each section of the MDA of the well. And during the flight, one of the astronauts would crank a turn on the MDA, causing the eggs to join.
Okay, so our procedure, the ITA put 125 microliters of dechlorinated water so that the frog eggs would be able to survive, was added to each MDA well, and 1 Xenopus laevis egg was also to be added to each well. And late loading and early harvesting was also requested in order to preserve the eggs as much as possible so that we could get the most accurate data. The control, like we stated, 2 eggs were left on Earth under the same conditions so we could contrast development.
Okay, so now once we sent our MDA, we set up a control group at the Florida Institute of Technology, where we didn't want to be shipping the eggs back and forth because we bred the eggs at the university, and so we set up our control group there. And what we were able to see is that through the same batch of eggs that went to the experimental group, because a frog lays many eggs, we were able to see that you can see automatically that we have development occurring with this. Now, on the experimental group, what occurred is, with all the delays on the shuttle, what happened was that they were only able to set up the top part of the experimental unit for the biological project, so we were only given enough room to put one egg in. And so with this one egg, when we received it back, we didn't determine any development, and so we don't know exactly what happened, seeing if it either was a dud or if it had problems, and it didn't really develop, and so we didn't really get much evidence.
Also, when we received the egg, it was white in color, and we were told from our breeders that this white egg meant that the egg was dead. So this began kind of what could we do to improve this. We kind of determined that it wasn't because of the G-force that our egg died, because obviously if previous experiments had been flown on Mach 6s, they had survived the G-force. So we didn't know if it was dead before it was loaded or if it was dead on the way home, but we also kind of concluded that it must have died early in development, because if it died on the way home, we would have at least seen some type of development, and we were unable to conclude any development from the egg. So some improvements. Obviously, it's so hard to be so distant from your project and not to be able to be there and select the actual eggs that are going on the shuttle. Obviously, if we were there, we might have been able to make sure that the egg was alive before we loaded it, just to be more in contact with our project, and also just closer to be able to make sure everything went right. And I know more contact during the flight to be able to observe possible development during the flight, to see if to also better manage our project that way.
So basically, we didn't really get much application, seeing that the control experiment worked just like a developing frog. Morphogens worked in their concentration just as usual with gravity. But seeing as we had a dead egg, we weren't really able to get much. We're sending it right now to the University of Utah, hopefully to get more further study to see if we can really answer this question of what happened. Because seeing how it didn't really develop, we don't really know. And so with the application, basically we were hoping that we would be able to utilize these morphogens to change this concentration to really affect how things develop, so we can enhance traits, change these abnormalities, and really help the environment.
And just in closing, we would like to give a big thanks to Canyon School District, Hillcrest High School, both the administration and the teachers, as well as the SSEP and our parents. So thank you very much.
Were there any other proposals that you thought of?
Yeah, we went through quite a few brainstorming ideas, but we really just narrowed it down to this one. We didn't submit multiple proposals as a group. This was our only submission.
Thank you.
So the experiment may not have yielded the results you wanted, but what did you learn about yourselves as individuals and as scientists?
I would have to say, when we were first down in Florida and they first scrubbed the first launch, we had to scramble all together as a group to re-get fresh frog eggs, and getting fresh frog eggs is really difficult. And so what happened was we had to order frogs, and we had to breed them ourselves in our hotel room. And so I think the scrambling and doing all of that was probably the biggest thing, because we were able to come together and work together and really figure out a way to solve a problem like developing frog eggs in a day.
Also with, obviously, you know, it's a whole trial and error process with science, and we have experienced that firsthand with having to get new eggs when the shuttle was launched, because it is a very time-sensitive project, as well as not getting the results we expected. We know that, obviously, when you're researching something like this, you have to try and try again. So,
if the eggs survive, what would have been the benefit?
Well, like I said in the application, if the egg had survived and we had seen that microgravity does play an effect on the concentration, like, the possibilities are really endless, because we could use more experiments to find more examples of how we can manipulate these concentrations to see if we can really change how things develop. And if we can, then we can enhance things, we can change abnormalities, we can help create better animals, better things, just from animals to humans, because of how microgravity, how morphogens are in everything that develops.
Thank you.
All right, thank you again for excellent presentation.
Someone else to hold that besides me?